Hello students, and welcome to today's video where I'm going to be talking briefly about infrared spectroscopy. And the reason that we're going to talk about infrared spectroscopy is that this is a way that we can go ahead and help us identify really what kind of functional groups are on different molecules. And the way that it works is that we're going to go ahead and use some infrared light, and we're going to go ahead and shoot it at a molecule, and what happens is that molecule is going to go ahead and block some of that infrared light. And by seeing which kinds of light it ends up blocking, it's going to indicate to us what functional groups are there. Now, how this works specifically is that we need to go ahead and think of infrared light kind of like we do visible light. And in visible light, we're used to having red, orange, yellow, green, blue, violet, and indigo. And that's kind of like the range of colors that we see. And infrared light is also going to end up having that same kind of range of colors. It's just that we are unable to see them with our eyes. And so in this particular process, what we do is we're essentially shooting a rainbow of light at a molecule. And what happens is that some of the colors don't go all the way through the molecule. So I want you to imagine that you're kind of like shooting a light through a filter. So an example, you're shooting a light through a blue filter that kind of absorbs the color blue. What will happen is that when you shine your light, you'll have the entire rainbow, but on the opposite side of that filter that's filtering out the color blue, you won't be able to see that color. And that's what's going on with infrared spectroscopy, that we're shooting our light at a molecule and some of that light gets blocked. And being able to identify which type of bonds blocks that light and read the graph and seeing which light gets blocked is the core of what infrared spectroscopy is all about. Now, to start us off, I'd like to go ahead and take a look at what is actually causing that light to get blocked. And what's causing the light to get blocked is that molecules are able to absorb energy, in particular light energy, when it comes at them. And they're able to absorb it in three different ways. They're able to absorb it by either stretching, and when I say stretching, I mean that the bonds between those molecules will actually stretch apart just a little bit, and they'll be able to hold on to energy that way. The second way that a molecule absorbs energy is that it can kind of bend just a little bit, and that's going to be called scissoring in the plane. The last one is just kind of like twisting around in space, and those are the three ways that a molecule can kind of absorb energy. And in infrared spectroscopy, for the most part, we're going to be concerned with this one right here, the stretching vibration. The reason is that the stretching vibration often gives us a very, very clear signal about what's going on with a particular molecule as it's absorbing that energy. And so what we're going to do now is we're going to go ahead and translate over to our first graph. And this is our graph that we're going to get when we have an IR spectrum. And I want you to take a note of a couple of things on the IR graph. This right here is relative transmittance. So this is telling us how much of that light, or those different wavelengths of light, are able to get through to the other side, to our detector. And this right here is our wavelength of light. And this is where we would go ahead and kind of associate that with our colors. So we'd say, that red is over here, and orange is here, yellow, green, blue, violet, indigo. that's what's kind of going on here. These are the different colors of light that are being either transmitting all the way or being absorbed. Okay. And what we can see here is that first, relative transmittance, this is saying how much of it got absorbed on the detector at the end. Okay, and we can see that in this first little bit here, and where we said our orange to red zone would be, that all of that light seems to go through. So that indicates that any kind of bonding that we would normally have in this zone isn't present. All of this light went through. And at this part, it says like, hey, no, something is blocking that light. And so we need to be able to determine or know what happens or what types of bond ends up blocking this light. And that's what we're going to go ahead and transition to right now. In particular, we're going to start with this one right here, and this is nonane. And nonane, we know this as a molecule that has essentially 9 carbons. It ends up having 20 hydrogens, and it'll end up looking like this, where we have 1, 2, 3, 4, 5, 6, 7, 8, 9 carbons, and it has 20 hydrogens. And I want you to take a look. This has a lot of carbon-hydrogen bonds, okay? Which is handy because this helps us identify the first little section right here. And this section right here is going to be right at about 3,000. And you should see that there's a very deep peak right here. It is both deep and it's a relatively thin peak. And this is where our CH bonds are. So typically when we have a long peak right here, that indicates that we have some CH bonds. Now, being that this is an organic chemistry section, this is going to be something that we're going to see fairly often. That we're going to see a large stretch in the 3,000 bond for a whole bunch of different compounds. Okay, and so this is our first one here. So this one, we could take a look at this graph, and what we'll notice is that there's this big thing at 3,000, and because there's nothing really else that's giving us a big indicator, we'd be able to say, all right, I am fairly certain that this is an alkane. And I can say that because it looks like it just has carbon-hydrogen single bonds, okay? And that's what it's telling us right here. Next is the same type of molecule. It again is a non, so it has 9, but we have an ene, which means that we have a double bond. And what we're going to notice is that there is a slight difference between these two. Okay, we still have this extremely deep line right here, but we should notice that we have this new little nub right here, as well as this new section right here where it kind of comes down here. And what's going on here is that this is indicating to us that we have a carbon-hydrogen bond. So it's still a carbon-hydrogen, but that carbon is going to be double bonded to another one. So what this is saying in particular is this is indicating that we have some carbon and it is bonded to a hydrogen, okay? And so that's what this particular area means. If we have a little peak that ends up coming right here, all right? The next one that we're concerned about is going to be non-ene, okay? And non-ene, I want you guys to notice that we have a peak that's a little bit further out. And this is the same thing like before, except that it's going to be a triple bonded carbon to a hydrogen. And that's what non-ene is indicating with this one right here. So if you see your main peak, and then you see a secondary peak like this one right here, that indicates that you have a triple bond somewhere in your molecule. The next type of molecule is going to be what's known as an alcohol. And this one has a different type of structure. So I want you guys to see that in this case, we have our 3000 zone, okay? And this 3000 zone, this is still our carbon-hydrogen bonds. We still have a lot of carbon-hydrogen bonds. However, we also notice that we have this new big dip that's happening right here. And this new big dip that's happening, and it's happening between like 3,500 and 3,300, this indicates that we have an OH bond. So anytime we see a big dip that's like this, that's going to indicate that we have an OH bond. This still indicates that we have the CH bonds. And then at this point, I want to go ahead and talk about this area right here, because this area starts to look a little funny now at this point. And what's going on here is this area is kind of like our fingerprint area, where if we were studying this in depth, we'd be able to possibly go ahead and look and see a little bit more closely to compare what those different molecules would be. However, in this class, we're not going to look at this area all too much. And what happens here is that this area includes some stretching, but it includes a lot more of this scissoring and this vibration. And since it's easier for us to figure out the stretching, that's what we're going to be working with. Okay, so the main thing for an alcohol is that you'll see this big wide area, and it should be between like 35 to usually around 3,000. You'll get some variance with these, but in general, you'll have this big wide dip, and that'll help indicate that. Next is a C double bond O. Okay, so this is another one. This is pentanone. And pentanone has a C double bond O. It has what's called a carbonyl group. That's when a C is double bonded to an O. So we notice first that we have our 3000 group. That's our CH bonds. But we also have this deep, deep one right here. And this deep one right here, this is indicating for us that we have a C double bond O. Okay. And when that's at about 1700, or I would say, in the 1700 range, that's going to be a good indication that we have a C double bond O. And that C double bond O really absorbs that 1700 color, whatever color ends up being there, it really absorbs it well. So we get a nice, deep, thin peak right there. All right, this is another example. This is one that we see, first of all, we see that we have a big, wide, broad thing, and it's between 3,500 and 3,000. So that means that we're going to have an OH stretch right there. We also see it dips down a little bit more into 3,000. That's showing, hey, we have that CH stretch going on, which is expected because we have a lot of carbons and hydrogens. Then we also have a C double bond O stretch right at 1700, okay? And since we have that, that'll indicate that we have both a C double bond O and an OH stretch. So that tells us that, hey, this one is not a carbonyl, so it's not going to be a ketone or aldehyde. It'll end up being a carboxylic acid. And this one ends up being exactly that. It has a C double bond O and an OH. Means it's likely a carboxylic acid. This next one here is going to be an NH group. And what's going to happen is at about 3,500, what you'll see is you'll see a little brief come down where it'll come down to either like 30% of it's blocked or 50%. And you'll see this come down where a little bit of it comes down. Not all the way. It makes a little peak here. And that's going to be because of NH stretches. All right. So NH stretches are going to be between here. Notice it's not anywhere near as wide and deep as the OH stretches, and it has some variability to it, but it's nowhere near as deep as the OH stretches. Again, this 3000 zone, that's our CH bonds. And then finally, we have our phenols, or our carbon rings. And what's going to go on here is that our carbon rings are kind of a special molecule. They show up in a weird way here. This is a phenol, so we have a phenol, so we have this big dip here that shows the OH. But the carbon ring, that structure, in general, these peaks that are right here, and they should be right around 1500. Sometimes they might dip all the way down to 1400. But if we have really, really deep peaks here, that's going to indicate that we have some sort of ring that's going on here. And these help to indicate like the C double bond C's, or the hydrogens that are also attached to those ones right there, that are in that special ring. And so anything that's in this zone right here that's really deep, that's not like the 1700, that's less than the 1700 zone, really should be an indication that we have some phenol that's going on there. Okay, that's going to go ahead and close us out for the lecture portion for this. Make sure that in your notes you're able to identify what all the different particular peaks mean, so that you'll be able to clearly and quickly identify what functional group is present. You'll end up getting a cheat sheet that's going to look very similar to this, where they are going to go ahead and tell you what kind of absorption bands that is going to be available for your test, both your final exam at the end of the year and the test that you're going to be taking in class here. And they end up telling you about where those are. So as long as you're able to use both this sheet and analyze the graphs themselves, you should do perfectly fine and well with this particular topic here.